So our second talk today is from Jay Serebrenik, who is a postdoctoral fellow in the lab of Dr. Shalom at the University of Pennsylvania and Children's Hospital of Philadelphia. And it's in this postdoc work where he's been able to develop a high throughput approach for direct manipulation and visualization of endogenous protein, which they have called scalable pooled targeting with a ligandable tag at endogenous sites, also known as spotlights. So Jay's used spotlights to study endogenous proteins capable of degrading various targets upon induced proximity, as well as different protein quality control responses. So this work has been supported through his F32 and K99 awards. Prior to his postdoc, he had earned a BS from Tufts University and completed his PhD at Yale, working in the lab of Craig Cruz on small molecule strategies to study protein quality control mechanisms in the Golgi apparatus. So with that, I'll hand it over to you, Jay.
Awesome. Thank you very much for that introduction. And also a big thank you to the organizers for having me on this awesome seminar. So today I'll be telling you about our work developing a technology to manipulate the endogenous proteome and using it to find effectors for induced proximity-based applications. So in our lab, we're really interested in studying protein function at scale using functional screens. Traditionally, we use CRISPR screens by perturbing a specific gene in each cell. And CRISPR and related technologies have been instrumental in enhancing our understanding of biology, but ultimately they perturb proteins only in one way in affecting protein levels for the most part, and they do so indirectly. We sought out to expand the scope of functional proteomics by developing a technology targeting the protein directly rather than the gene. With this enables new ways to study proteins in high throughput pooled formats, where we can perturb these proteins in different ways, including by inducing changes in localization, affecting the protein levels directly, studying the protein localization patterns, and much more. So the way we do this is through a technology called spotlights, which stands for scalable pooled targeting with a ligandable tag at endogenous sites. And I'll tell you first what it does, and in subsequent slides, how it does it. So in short, we're able to generate a cell pool where in each cell we have a different endogenous protein tagged with a ligand binding domain, and we like to use halo tag. And then we can treat these cell pools with generic small molecules that bind the ligand domain, and it can induce an effect on the tag protein. So if we use a halo tag fluorescent ligand, we can look at the protein localization patterns. A destabilizer will induce protein unfolding, a degrader will induce degradation, and so on and so forth. So I'll begin with kind of the punchline and show you just how these libraries look like in real life. And so here's one of our libraries that was treated with a fluorescent ligand. And you can see from this one image, there's a huge variety of different localization patterns. And I'll just point out a couple to you. So up here, we see a pattern that looks like mitochondria localization. Below that, we see what looks like a nuclear membrane pattern. Nearby, we see some patterns that look like could be cytosolic bodies. We see lots of localization patterns throughout the nucleoplasm, as well as the nucleoli. We also see the cytosol. And in the bottom here, we see what look like Golgi localization pattern. And then near that, we see the plasma membrane. In the center, we have a pretty nice localization of the cytoskeleton. And we see the ER, and then farther off to the right, we see some kind of nuclear bodies. So you can see that these libraries really can be quite diverse. And to note again, these are all endogenous proteins that are tagged here. So this talk is divided into two parts. One, I'll tell you about how we make these libraries. And the second part, I'll tell you about how we use them to perform induced proximity screening. And you can find more details on these projects linked here below. And I also really want to emphasize my wonderful and super talented teammates that have worked on this project, including my co-first author, Steph Sansbury, and Dr. Tomer Lapidot, who have helped in the development of spotlights. And Deepak Mani was instrumental in performing the endogenous protein recruitment screens. And both of these projects were done in close collaboration with the wonderful lab of George Burslem here at Penn. So despite the huge advancements in CRISPR technology, it's actually really difficult to tag proteins in a scalable fashion. So traditionally, endogenous tagging is done with laborious and costly methods such as homology-directed repair, which requires specific reagents to be developed for each targeted site. Our method, which we published a few years ago, instead relies on homology-independent integration. And with this approach, we only need a site-specific sgRNA, and the rest of the reagents are completely generic. And we can use them for any targeted site. So we create a double-strand break with the sgRNA, which is filled in by the DNA cassette, and our tag is surrounded by splicing sites. And after transcription, the tag is spliced in seamlessly into the final product. So you'll notice we target introns, and that is for a few different reasons. One is because homology-independent repair-based approaches are prone to indels, which are less disruptive in introns. So we basically insert a synthetic exon, which gets spliced into the final product the same way, regardless of the indels that we create. Introns also tend to be quite large, so we have a large variety of spacer sequences to choose from. And then another reason is that we're able to add selection markers into our DNA cassette that can help us select for genomic integration events, but because they're outside the splice sites, they don't actually get spliced in the final product. So with this approach, we get really high efficiency tagging, requiring only a site specific guide RNA in addition to the generic components. So we can use this approach to create pooled tag libraries using an approach that's very similar to CRISPR screening, where we can create a lentiviral pool of sgRNAs, transduce cells such that each cell gets only one sgRNA, and then transfect the cells with the generic reagents, including the donor, the guide to linearize the donor, which is also generic, and Cas9 to perform all the cutting. Then we label the cells with a fluorescent halotag ligand, and the cells where there are productive fusions and viable fusions, they'll be brightly fluorescent, and we can enrich for these cells by fluorescence activated cell sorting. And so we end up having a very diverse library with lots of different proteins tagged. Not only do we tag lots of different proteins, but we can also tag the same protein at lots of different sites. So genes on average have about 9 introns, and all of our libraries utilize as many introns as we possibly can for each gene. I also want to emphasize, and this might be obvious, although we're tagging genes internally, this doesn't mean that we can't find sites on the surface of the 3D protein structure. So once we generate these large libraries, because all the cells have an integrated sgRNA, we can analyze these libraries using sequencing-based approaches. And sequencing will, the sgRNA will not only tell us what protein is tagged, but exactly where the protein is tagged. So we use bulk sequencing to assess the quality of our libraries. We look at the success rate at the level of the guide and the level of the gene. And on the gene level, our success rates are quite high, approaching and oftentimes exceeding 90% of our intended targets. The tags are, we see a large variety in the tags. So lots of different compartments. There's no strong biases in that sense. So what kind of genes, proteins we can tag. We also assess for the recovery of essential genes, and this gives us an idea as to whether tagging the tags that we enrich are functional fusions. Because if tagging was disruptive, it would be very difficult to recover tags against essential genes. But the reality is that it's not, which suggests that for the most part, the tags that we recover are functional. We can also study these libraries by optical sequencing, and this is a method developed by Paul Blaney's group a few years ago, where we can essentially both phenotype and genotype the cells on the same cover slip and get the information for each cell of what protein is actually tagged and where it is tagged. So with this, we can assess localization patterns for all of our tags and get an idea of the expression levels as well. So I'll quickly go over the protocol. We basically just plate cells on in dishes, stain them with a fluorescent ligand, fix them, perform high magnification phenotyping, and then we amplify the sgRNA sequence and perform at a lower magnification sequencing by synthesis to get the sgRNA sequence for each cell. Then we overlap all of these images, and then we're able to tell what gene is tagged for each cell. And so in the process of the image analysis, we segment the cells and we can generate this really nice albums at the level of the guides, at the level of compartments. And this gives us another way to assess the quality of our tagging. So in our initial libraries, we focused on genes that had well-established localization patterns. And so simply by looking, seeing where they distribute, we can see did the tagging lead to an expected distribution pattern or not. And so here are some examples of some tags that work for all these different compartments. And all of these compartments are represented by multiple different proteins tagged at multiple different localizations at sites in the gene. So in the ER, for example, it's represented by calreticulin and calnexin, and both of these genes are tagged at completely different sites, yet they all have the expected localization pattern. So we did a full quantification of these localization patterns that I won't show to you, but basically we see the vast majority of our tags having the expected localization patterns as opposed to not. So once we generate these large, comprehensive, diverse, and functional endogenous protein tag libraries, we can do lots of really cool experiments with them. One of which is to do induced proximity screens, which I'll talk about for the rest of the talk, except to mention really quick, another really cool application we do, where we treat these libraries with a hydrophobic tag. And this induces a spatially restricted protein unfolding, which allows us to systematically study proteostasis responses throughout the cell. And you can find more details on this work below. So in studying effectors, we're really interested in exploring effectors in a completely unbiased way. So for a given target degradation task, rather than explore the handful of different effectors that are currently available, we instead are interested in testing every single endogenous protein in our cell model of choice, each with multiple different orientations to the target, and thereby find the perfect effector pair for the degradation task. So this approach allows us to find effectors that might be better at degrading specific targets. Maybe a target is not capable of being degraded with the existing effectors, and we can try to find an effector that would work against it. We could find effectors with better cell essentiality profiles, and these effectors might be less prone to leading to resistance mutations, which we know are an ongoing concern in existing clinical trials with degraders. And we can also find effectors with better cell expression profiles that might have activity only in the cells and tissues that you're interested in, not in the ones that you're not. So we tried this first, not with quite the entirety of the proteome, but with over 900 proteins associated with the proteostasis machinery, including members of the ubiquitin proteasome system, the autophagy lysosome pathway, and various chaperones. So we generated the Spotlights library in the manner that I described previously. And here's an image where we stained the cells with our fluorescent halotag ligand, and we can see a variety of different localization patterns. And each cell in this library is also expressing M-Clover FKBP, which we use as a model target and to help us optimize conditions for performing these screens. So to figure out what ligand to use and at which concentration, we overexpressed an effector that was shown to work by overexpression Parkin, and we fused it to mRuby to also be able to detect induced proximity and ternary complex formation. This effector, this system was able to degrade our target, but only slightly, which was actually quite nice because we were able to measure then degradation and induced proximity in the same system. So the results are shown here on the right. There are two y-axes in this plot. On the left, we're looking at M-Clover fluorescence intensity, represented by the solid lines. And on the right, we're looking at the Fret signal, represented by the dashed lines. And the different colors represent ligands with different linker lengths. And so just summarize the conclusions, we see that our longest ligand is the one that is able to induce the strongest degradation and also have the strongest Fret signal. So this is the ligand that we used for our screens at the concentration indicated from the experiment. So we performed two orthogonal screens against two different targets with two different ligands. One was our M-Clover model, which was overexpressed in these cells. And then another, we looked at endogenous BRD4. So for the M-Clover screen, we took advantage of fluorescence to enrich for viable effectors. So on recruitment, an effector that would lead to the degradation would lead to a reduction in fluorescence intensity levels in the cell, and we would enrich for these cells by fluorescence activated cell sorting. For screen BRD4, we took advantage of the fact seen in our lab and in other studies in the field that BRD4 degradation is much more toxic than BRD4 inhibition. So we screened for effectors that, when recruited to BRD4, led to degradation and led to cell toxicity. So this was a growth screen that over time, the viable effectors would be depleted from the population. And then on sequencing, we'd be able to identify what these are. So the results are shown here. On the left, we have the results from the M-Clover screen. On the right, from the BRD4 screen. On the x-axis, we have the normalist counts for each different tag. And on the y-axis, I won't go into details, but it's basically the magnitude and direction of the effect. And what I want to draw your attention here is that despite the fact that we have two very different screens targeting different targets with different ligands, both of these screens actually converged onto the same top hits. And these hits are WDR26 and RAMBP9, which are components of the CTLH-E3 ligase complex, which we were really excited to see because there's some evidence that members of the CTLH complex are upregulated in certain cancers. And there's, in some specific cases, some of these components are thought to be even cancer drivers. So this, in the right context, these could be ideal effectors because they would have a good essentiality profile, where they might be less prone to developing resistance mutations, and because they're overexpressed in these cancers, they might have a better tissue specificity profile as well. So keep on the lookout for further studies on these proteins from our lab. So we validated that they work against these targets. We first looked at M-Clover by doing flow cytometry experiments. We created clonal cell lines where we tagged WDR26 with halo tag. And here's looking at some single cell analysis where cells treated with DMSO are shown here in blue. Cells in green are treated with DTAG V1, our positive control that engages the endogenous VHL E3 ligase. And then in orange are cells treated with the halotag FKBP ligand. And for WDR26, we see extremely potent degradation of M-Clover that is the same as DTAG V1, and in certain experiments actually can even exceed it. RAMBP9 is also quite potent, but not as strong as WDR26. We performed some dose response curves where we see a hook effect and some time course experiments as well. And we see WDR26 can induce almost complete degradation in under 24 hours. And RAMBP9 also has increasing degradation as time goes on. So in some experiments, we perform just longer incubations with the RAMBP9 effector to get a stronger effect. Similar story for targeting BRD4. So here's a blot against BRD4 in the top row, and WDR26 recruitment induces very potent degradation at a wide range of concentrations tested. M-Clover is only degraded with the halotag FKBP ligand as opposed to the halotag BRD4 ligand. And as a negative control, we recruited, we used an effector that was not a hit in the screen to recruit to BRD4. And in these cells, this did not affect BRD4 levels at all. And in the parental line where nothing was tagged, that obviously had no effect as well. The chart on the right emphasizes the potent toxicity we see with WDR26 recruitment. Recruiting WDR26 as opposed to the inactive effector, we see after 72 hours of treatment over 3 orders of magnitude in difference in the cell count. So the effect was really strong, which is why the results of the screen were turned out to be quite nice. Finally, we looked at the mechanism of degradation for both of these effectors. We first looked at whether they were dependent on the proteasome by inhibiting it with MG132, and we see that both WDR26 and RAMBP9 are dependent on the proteasome. We see that with vehicle control, we see decent degradation, and then with the proteasome inhibitor, that gets reversed. And we also looked at the dependency on other CTLH members for these effectors by using CRISPR knockout screens. In our non-targeting controls, we see strong degradation for both WDR26 and RAMBP9. Each point here for these bar plots is a different sgRNA for knockout, not just a different technical replicant. And so we see that knockout of the effectors themselves, we see an inhibition of degradation. And these effectors also depend on the catalytic core of the CTLH complex, namely MAEA and to a lesser extent Armin D5A. So it was nice to see that these effectors were working kind of how we expected them to. So to conclude, I've shown you how these recruitment screens are really flexible and really powerful ways to study effectors. And I'm really interested in expanding this to looking for effectors for any kind of protein modification, not just degradation, but also protein stabilization, disaggregation, and any kind of post-translational modification. And this approach is really amenable to look for all these effectors because we simply create these libraries and after recruitment, just have some kind of way to enrich for the desired phenotype. Here I showed you fluorescence-based and growth-based approaches, but we could really adapt these screens to for any kind of to read out any kind of phenotype sequence and then identify these effectors. And this could really help discover new biology and lead to the development of new therapeutic modalities. So in summary, I've shown you that our Spotlights technology is capable of creating large, comprehensive, diverse, and minimally disrupted libraries. And we use these tools to perform screens to discover new effectors for targeted protein degradation, discovering members of the CTLH complex as potent members of degradation. We're really excited and think these tools have the potential to be really powerful and flexible approaches to study proteins and for the development of new medicines in the future. So I want to thank you all for your attention. And I want to thank my lab as well, especially my fantastic advisor, Ophir Shalem, and my wonderful, talented teammates, Steph Sansbury, Karna Lapido, Deepak Mani, and our great collaborators at the George Burslem Lab. This work was supported by an F32 and the K99 from the NIH, and I'm currently on the job market, so maybe you will see me there soon. So thank you very much, and I will take any questions.